Hello, I'm Gary Schoenwolf from the University of Utah, and I'm delighted to be here today in the midst of January with Bruce Carlson. We're in Tucson, Arizona, where it's about 70 degrees today. Bruce is from Minnesota, and I'm from Salt Lake City, Utah, so it is much nicer weather here today. Bruce has agreed to share some insights he has gained during his 40-plus years as a faculty member in anatomy at the University of Michigan. Bruce, as I've said, is now living in Minnesota and Minneapolis, and I'm sure he's equally appreciative of being in this warm climate as I am today.
Here, here.
And very much enjoying the weather. Welcome, Bruce. I'd like to begin by providing a brief overview of Bruce's career. We'll go all the way back to your birth, Bruce. Bruce was born in Gary, Indiana, and then throughout his education, he spent all of his time in Minnesota. He went to high school in Minnesota and then received a Bachelor of Arts degree, summa cum laude, in biology, languages, and chemistry at Gustavus Adolphus College in St. Peter, Minnesota, and then went on to get a Master of Science degree at Cornell University in Ithaca, New York, where he studied ichthyology, embryology, and histology, and then an MD, PhD degree at the University of Minnesota in anatomy and zoology. There are two themes that I think are very unique to Bruce's career that I want to explore with Bruce today. He has a wide variety of interest, and this is the first theme. His career began in fish biology, and then he moved to developmental biology and anatomy with a focus on regeneration, first in limb regeneration, and then moving to muscle regeneration. And the second theme is during the Cold War in the 60s and 70s, Bruce spent much of his time in Russia and in an East Bloc country, Czechoslovakia. So I'd like to explore those two themes this day and some questions that I want to ask Bruce. So Bruce, you started out wanting to become a fish biologist. How did you make this transition from fish biology to developmental biology and eventually on to regeneration research?
Well, I was always interested in fish and fish biology and had already committed myself to taking on a master's program at Cornell in ichthyology. And the last day I was at the small college, Gustavus Adolphus is the way we pronounce it.
Okay.
Yeah, it's a small Swedish school. My being Swedish, it's reasonable that I would go there, I guess. But the last day I was in college, I went to the library, and they had just started subscribing to the Journal of Experimental Zoology. And the first issue they had contained an article by Richard Goss and Martha Stagg on the regeneration of the lower jaw in newts. And I read the article, and for me it was almost like a religious conversion. I said I wanted to do regeneration for my career. So I went to Cornell, told my advisor that I still love fish, but I really ultimately want to do regeneration research. And he says, go ahead, if you can do both, that's fine. So I spent about 20 hours a week during the time I was at Cornell reading every article on regeneration in the Journal of Experimental Zoology while doing my fish work. And then after that, I basically went to medical school and graduate school too.
Oh, very interesting. I should point out that at the time, probably the Journal of Experimental Zoology was the leading journal in developmental biology.
Yeah. Certainly in this country, yes.
In this country, definitely. And so you were really reading the top literature in the field.
Yeah.
So what is also very unusual about you is that you made this move of conducting research in Russia, and this was extremely unusual at the time of the Cold War. What led you to go to Russia when you chose to do so?
Oh, this is another long, very circuitous story, but one of my other major interests is in languages, and one Christmas vacation when I was in college, I was in a used bookstore, and I happened to see a Russian-English dictionary for sale for 50 cents, and I said, this sounds like a good deal, I'll buy the dictionary. So I bought the dictionary, didn't use it until I was at Cornell, and I decided to take a Russian course so I could get my money's worth out of my dictionary, all 50 cents. So I took a Russian course, scientific Russian course at Cornell, And then another incident involving a used bookstore. They had one at the college there at the university at Cornell. And one of the books that they had was a very unusual Russian symposium volume on regeneration that was written in 1953. And by that point, I could read a little bit of Russian, not very well, but I was going through this symposium report, really, and I could understand most of the language, but I couldn't understand the introductions and conclusions, which were all about trying to purify Russian science against what was called Morganism, Weissmannism, Mendelism. And it really didn't make much sense to me. So I put that aside, and then later on, after I'd been in Minnesota for a while, I took a little more Russian in Minnesota. And when I was doing my final oral exam for my PhD, I had gone through the usual committee interrogations, and they sent me out to the hall to wait tremblingly, so to speak, for the verdict. And while I was there, I opened up a copy of Science magazine, which had an announcement of the first exchange between the U.S. and Russian Academy of Sciences. And I got so excited at the time that I went to my lab and started writing a letter to apply for this exchange program. And then my committee chair came out and couldn't find me, so my committee went around looking for me, telling me I had passed my final oral. But that's basically how I got to Russia. And the day I was supposed to graduate from the University of Minnesota, I was in an airplane heading for Russia.
My goodness. So this was a jointly sponsored program by the two governments?
Yes. The U.S. had this with Russia, or the USSR initially, and then they had similar programs with many of the Eastern European countries.
Yeah. Okay. Very interesting. So what was it like being in Russia at that time? Very interesting competitive time between the two governments.
Yeah, it was actually fascinating. When I was there, I was working in the Institute of Developmental Biology in Moscow, and no one there had ever seen an American, and of course I had never seen a Russian. And I still have a vivid memory of my first day in the laboratory. I was sitting at my desk, and there were a couple of bookcases like this with a little crack between them. And I looked between the crack to see who was sitting at the next desk, and the woman there was looking between the crack, looking at an American. So we both realized that we were both curiosities of a different sort. But life there was, let's say, interesting. It was not for the faint of heart at the time. Living conditions were a bit, say, spare. One place, I stayed in many hotels. And one place I was at was an old Russian apartment building that had been converted to a hotel. And I was in a little room that I think might have been the kitchen or something like that. But for the first week I was there, every time I came back from work, my trunk that I had was submerged in about 3 inches of water. So I finally learned to put my trunk on top of the bed until they got the pipes fixed. And food was not the greatest in the world. And of course, in that era, you assumed that every room was bugged, and there were all sorts of people who'd be following one around. And the Russians always knew where I was if they wanted to get a hold of me. And had many experiences, especially with KGB types over the years. And I could write a book or two about that.
Maybe we can come back to that at the end and talk a little bit about that if we have time. That'd be great. So were you by yourself at this time?
Yes.
Or?
Yeah.
So Gene wasn't with you yet? Okay. And how old were you roughly at this time?
I think I was 27 when I finished school.
Okay.
Yeah.
Very cool. So you also then went on to conduct research in an East Bloc country in Czechoslovakia. What caused you to do that?
Yeah, well, that was also interesting. And by that point, I had expanded my research from limb regeneration to muscle regeneration, and we can probably talk about that a bit later. But I was on a 9-month appointment at the time, and I had a pretty good research grant, but it wouldn't pay summer salary. And I had the option of teaching histology to flunked-out medical students or doing something else more interesting. So I applied for an exchange fellowship between the U.S. and Czechoslovak Academy of Sciences, and they accepted me for that. And I wanted to work with a fellow who was, at the time, doing work on muscle and culture, looking at what was a very popular topic at the time, amitosis, or amitotic cell divisions. And it turns out that the Czechs wouldn't let me work with him because he was in a military institution. So I was given my second choice, which was Ernest Gutmann at the Institute of Physiology of the Czech Academy of Sciences. And he and I got along like ham and eggs. He was an absolutely brilliant scientist, and he was really my mentor, because I never had a mentor when I was a graduate student because he had died. So we were there, my wife and I, this is around 1971, we were there for a whole summer, 4 months in the beginning. And after that point, my research, by the way, and I don't think I'll talk about this until we get into some details of research, but it wasn't going well at all for most of the summer, and it wasn't until the last 2 or 3 weeks that things really clicked, and clicked so well that I decided to go back. So every year during the 1970s, I spent at least a month in Czechoslovakia conducting research both at the Institute of Physiology and at the Charles University in the anatomy department.
Mm-hmm. So how did life differ on a day-to-day basis between the Soviet Union and Czechoslovakia at that time?
One of the major differences is, well, a couple really strike me. One of them is the atmosphere. In Russia, I felt very comfortable personally there with the Russians, particularly because we were both big countries and we had the defects of people who live in big countries. With Czechoslovakia, I always felt like I was a person in a big balloon waiting to get pricked, because they have a different mentality when you're under the thumb of somebody else. Okay. But the dominant thing that I felt there was fear, because this is a couple years after the Russians had moved in and squashed the 1968 Prague Spring. And the institute I was at was one of the prime movers in terms of trying to liberalize Czechoslovakia. So they were stepped on very heavily by the government. And I remember one time when I was working, there was an announcement over the public address system saying all workers in the institute must come to the auditorium. You know, it turned out that a member of the Czech Central Committee was there basically blasting the institute, and little did he know there was an American sitting in the front row, much to the amusement of everybody else in the institute, by the way. But they had this way of doing things where it's called Schweikism, where you try superficially to obey the rules, but in reality you're subverting everything you possibly can. But that was life in Czechoslovakia.
Was there a strong military presence on campus, or just not really?
Well, where I worked in Czechoslovakia, it wasn't a military presence so much as they had guards. The institute was surrounded by a fence, and I had to show a particular pass in the morning, but and the director of the institute actually often used to look out the window with binoculars to make sure the guards were doing their thing. But after about a week or so, I started using my Bank of Ann Arbor pass, and they let me in on that every day, so they didn't look at it too closely. But one of my colleagues actually was working late one night, and the door was closed, so she jumped over the wall, and they shot at her. So it was, it wasn't all benign.
No, I guess not.
Yeah.
Well, that's fascinating. So you began your developmental biology career. So this is the kind of the post-fish life.
Okay.
But we'll come back to the fish life, certainly, with limb regeneration.
Yeah.
And then you made this transition to study muscle regeneration. Yeah. And it's something I've always been curious about, why you made that transition, because limb development is so fascinating, of course, and limb regeneration is very fascinating. So why did you make that transition?
I never really gave up limb, and I find limb development still absolutely fascinating, and I continued that for probably 20 years or so. But when I was in Moscow in 1966, I became acquainted with a professor named Alexander Nikolaevich Studitsky, who had...
It's a mouthful.
It certainly is. Fascinating guy, who had come up with a new model for producing muscle regeneration. And his background, at the time in the 1960s and for the previous 50 years, the dogma was that skeletal muscle cannot regenerate. And this was in almost every histology book, every surgery book. And a few people, about once every 10 years, somebody would write a paper on muscle regeneration, but it was generally disregarded. But Studitsky, who had worked on both bone and muscle since the 1930s and did some absolutely brilliant biological work at the time, he was also a very politically attuned animal. And in 1948, he was working, as all the other Russian biologists, during the time of Lysenko. And maybe I should interrupt by giving you some background of this, because otherwise what I'm going to tell you won't make any sense. Lysenko was a Ukrainian agronomist who was rather ill-educated, again, a highly politically tuned animal, who decided early in his career that the environment trumps heredity, and did some experiments involving what they called the vernalization of wheat. And I won't get into those details, but the basic idea was that classical Mendelian genetics is really an artifact and should be eliminated. And he got Stalin's ear, and during the 1930s, he began a purge of classical Russia genetics. And by the end of the 1930s, he had taken a Russian plant geneticist, along with hundreds of other geneticists. But this fellow, his name was Vavilov, and he was probably the preeminent plant geneticist in the world at the time. And he had him thrown in jail, and Vavilov starved to death in prison a year or two later. But under the influence of Lysenko, literally a whole generation, or actually 2 generations of Russian geneticists lost their jobs, and the whole discipline lost its standing in the Soviet Union. But a sub-cult, and I call it a cult, under Lysenkoism was something else which is even more bizarre. And this was headed by a woman who I think had a high school education, but certainly no more than that. And her name was Olga Borisovna Lepeshinskaya, speaking of mouthfuls. And she had the idea, she came up with what was called, in English we would call it the new cell theory. And her idea was that cells do not have to arise from pre-existing cells, but instead they can really form from what she called a living matter. And this would happen both in embryogenesis and in regeneration. And she published several books on the topic. I actually had them in my library before I gave them away. But she also was sycophant of Stalin and got his ear. And by 1948, her new cell theory was considered to be the official biological doctrine. And I've talked to a number of people my age or a little older who studied that new cell theory in both the Soviet Union and Eastern European countries. It was obligatory to follow that line. And that continued, I think, until 1953 when Stalin died. And then this particular component of Lysenkoism also died out. But in 1948, getting back to Studitsky now in muscle, he was, at the time, a believer in the new cell theory. And whether he really believed it or not, I don't know. But he did an experiment where he wanted to prove the validity of this theory. So he took skeletal muscle, which at the time everybody knew couldn't regenerate and had no capacity for cell division. And he says, I'm going to take a muscle, chop it into very small pieces so that there could be no surviving cells. And then if I get a new muscle growing from this chopped up muscle material, it'll prove the validity of this theory. So he took the chicken biceps muscle and the rat gastrocnemius muscle, chopped them up into material. He called it kakasha, which is Russian porridge, so it really finely mashed up, and put this tissue back into the arm of the chicken or the wing of the chicken or the leg of the rat, and wrote a paper, and I think in 1949 was his first paper, saying that it, as a matter of fact, did reform a new muscle. And then in 1953, going to this symposium volume that attracted me to Russia, he talked about these experiments. In his introduction of his paper, spent about 2 or 3 pages talking about the new cell theory, condemning Morganism, Weissmannism, Mendelism, and then after the obligatory political part of his article, then he got into the science. But during the 1950s and early 1960s, his laboratory did a huge amount of work on muscle regeneration and muscle transplantation. And even though the basic theory was crazy, he got some remarkable results, and they did a lot of work on nerve-muscle interactions, did a little bit of early work on contraction of regenerating muscle, but that wasn't horribly good. But basically set the stage for this whole thing. And I visited his lab in 1966, and it was fascinating because the first things we talked about was muscle from extinct animals. And whenever someone would uncover an animal from the Siberian tundra, they would send muscles to him. So I saw muscle of Siberian mammoths, musk oxes, and whatever else they happened to dig out of the earth. But then he showed me his muscle regeneration experiments, and when I looked at it, I realized he had discovered something remarkable. But nobody, first of all, he never published in English, and nobody could have access to the journals. He went to one symposium in, I think it was 1962, and his English wasn't very good, and nobody could understand either his English or his...
outside the country.
Yeah. I think it was in Hungary or someplace like that. Okay. But anyway, and several people from England were there, and I've talked to them, and they said they had no idea what he was talking about. So anyway, when I came back to the University of Michigan, then I decided to see if I could replicate his experiments. So for about 2 years, I I worked first on frogs and then on rats and chopped up muscles, put the muscle tissue back in the limbs, and sure enough, they did regenerate new muscles. And remarkably enough, in the rat, they would form proximal tendons, distal tendons. They had a nerve supply, blood supply. They looked like a generic muscle, but they didn't look like the original one. And this gets into some complexities of morphogenesis that I won't get into right now. But anyway, it worked. So I wrote a few papers on that.
And you probably need innervation for that regeneration to occur?
Yeah, it gets very complex. Early regeneration, what happens in these minces is that all of the muscle fibers die. And by the way, this is something else for the non-muscle people. In 1961, the satellite cell was discovered, which is a stem cell of muscle. And Studitsky knew nothing about these when he did his experiments. And when I did my experiments in the mid-1960s, no one still really connected much satellite cells with regeneration. There was a little bit of work in that area. So anyway, when you put this massive minced muscle in the limb, it all goes into a condition of ischemic necrosis. But then blood starts growing in, or blood vessels start growing into this mass. And as they grow in, macrophages would enter the mass of muscle. And then you would look, and at the histological level, the outer tissues would start becoming basophilic, and you'd see spindle-shaped cells, or myoblasts. And the inside of the muscle was still dead as a doornail. It was just a light pink necrotic tissue. And by the end of about a week or week and a half, the entire muscle was vascularized and was full of regenerating muscle fibers. And there was a gradient from mature cross-striated muscle fibers on the outside to very simple myoblasts or myotubes on the inside. But anyway, it really did work.
And nerves then don't play a role until you get myotubes?
Yeah. And this is the interesting thing, because in limb regeneration, if you don't have nerves, you won't get regeneration at all. In mammalian muscle regeneration, the early stages are totally nerve independent. And there are species differences. So in a rat, if you don't have nerves, the muscles will form cross-striated fibers and then gradually atrophy. In a mouse or a frog, if you don't have nerves, by 21 days, everything has degenerated and disappeared. But basically, the early stages are nerve independent. The later stages require nerves. Yeah.
Oh, very interesting. So I was going to ask you what was the most unexpected result in your research career. Yeah. And you've sort of given me a result that I think most people at the time would have said was unexpected. It probably wasn't unexpected to you.
No.
Because of your experience in Czechoslovakia. Yeah. But are there other cases where you had unexpected results that sort of changed the direction of your research?
Yeah, absolutely. And interestingly enough, it occurred at the same time in the 1960s. When I was a graduate student, I was interested in inhibitors, seeing if I could inhibit limb regeneration. And I was able to get some early samples of actinomycin D, which, at that time we thought it was a big deal that it inhibited DNA-dependent RNA formation, which at the time was, they were just doing the genetic code at the time. So it was a fairly advanced drug for the time. So I injected this into salamanders, and of course it stopped regeneration, but it had horrible side effects. This was initially in newts. So then after I did that at Minnesota when I was a graduate student, is something in addition to my thesis work. But then when I got to Michigan, I put together an axolotl colony, and I went back to actinomycin and decided to see if I could eliminate the side effects to the whole animal. So what I did is took the skin off the axolotls and soaked the skin in actinomycin D, and then sewed the skin back on, and then cut the arm off. And these things had a little bit of inhibition of regeneration, but not a great deal. But at least we didn't have the side effects. But about a year after I did these experiments, I looked at some of these animals, which I hadn't paid much attention to. And a couple of them had arms that were sort of like this instead of like this. So I got to thinking, well, maybe I sewed the skin on backwards. So trying to see if I could reproduce the backward arms, I took some axolotls and took the skin off from about here to here, and just rotated the skin 180 degrees and sutured the skin back, let it heal for a few days, and then cut the arm off. And much to my surprise, I didn't get arms like this, but they regenerated limbs, but the limbs were double or triple with up to 15 or 16 digits instead of 4. So that was a rather remarkable result. That was definitely unexpected, yeah. So then I continued for about 5 or 10 years along that line, looking at what might control this bizarre morphogenesis.
Yeah. Well, that's very interesting. So we often have unexpected results that change our own programs.
Yeah.
Are there papers of yours that you published that you think have changed the field dramatically? Or another way to say that, if you never existed, would the field be the same? Which is an unfortunate way to put that for any of us who do research.
Yeah, I mean, frankly, muscle regeneration would have continued without me, because soon in the 1970s, with the big controversy was satellite cells, whether muscle dedifferentiates or differentiates on the basis of satellite cells. I think the work I did demonstrated the capacity of muscle to regenerate huge amounts of information. And when I was in Prague, we also, because minced muscle regeneration didn't work very well for small muscles, we got into what we called free muscle transplantation. And Gutmann and I did this initially in 1972, and it turned out to be much better for certain experiments than mincing the muscles, and it was the same phenomenon. The muscle degenerated and then regenerated. But at the same time, an English surgeon named Noel Thompson published a first report of transplantation of a muscle in humans, a human with facial paralysis. And he was a classical surgeon, never in his life believed in muscle regeneration. So what he did is took a muscle from the foot of a patient, denervated it for 6 weeks, and then transplanted the muscle to the face to try to keep the mouth from sagging down. And so he had the muscle going from here to here. And this got the European plastic surgeons very interested in muscle transplantation. So I have continued. Actually, I was at my last muscle transplantation meeting a few months ago, but they were very much interested in this phenomenon, and so I spent a lot of time in the early 70s with plastic surgeons in Europe working out techniques. But then what happened in plastic surgery is that their version of microsurgery came into being, and they could, instead of doing what I call a free muscle transplant, putting the muscle in place without a blood vessel connection, they could do a vascular anastomosis. And the minute they did that, they converted the model from a muscle regeneration model to a transplant model, where regeneration was irrelevant other than the nerve, which is still a huge problem. So at that point, my interest in transplantation sort of veered away from theirs.
Very interesting. So I have to ask you this question, since I'm a developmental biologist, and I still have an active lab, and I'm still looking at problems to solve. So the question I have is, developmental biology has changed dramatically, as you know, over the course of your career, starting out primarily as a descriptive or morphological science, and now moving to molecular biology and genetics, so molecular genetics. So what do you see as some of the most important questions that still remain to be answered? And you could take that very broad, or you could make it very restricted to muscle, limb regeneration, whatever you want to do. But I'd be curious about your thoughts.
Yeah, well, I think I'd probably focus on regeneration. And interestingly enough, some of what I consider the most important problems in the future are some of the oldest problems that were ever posed in the field. And the one that still drives me mildly nuts is the experiment that drove Hans Driesch nuts 100 years ago or more. If you take an early embryo, a 2-cell embryo, and cut it in half, and it regulates, is it because the one cell knows that the other cell is missing, or doesn't it know that the other is missing and thinks it's the whole object? And you could take this to a more complex version of adult structure. But the same question exists, and I put it in what now is considered by many people an outdated concept of morphogenetic fields. But we still really have no idea what the molecular basis of form is. We're beginning to just touch a little bit onto it in terms of regeneration. Much more is known in the embryo. But in terms of limb regeneration, it's really a black box. So the molecular basis and cellular basis accounting for form is a really major one. Another one that relates to that is this phenomenon that I call positional memory, which gets back to the skin rotation experiment. When you rotate the skin, there's morphogenetic information in the skin and also in skeletal muscle, by the way, which remains, and it's very persistent. It even persists through degeneration and regeneration of the tissue. And it has a demonstrable effect on the regenerate. We have no idea what positional memory is at a molecular basis, but yet it's obviously very important.
Would you speculate in today's terms of how we think about development as maybe being epigenetic?
I don't know if you would call it epigenetic. The thing we always were thinking about is something on the cell surface. And of course, what the basis of that is, it could easily be epigenetic in the molecular sense. But I think it probably is going to get down to maybe some common molecules, what we already know. These days, people aren't finding many new ones anymore.
That's true.
But nevertheless, there are a few. There's one molecule in the limb, it's called PROX, which has a very pronounced effect on proximal distal morphogenesis. So we're at the point where at least we know something in that area. But in the cross-sectional axis of the limb, we know virtually nothing. So that's a significant problem to be solved. Another one, which is from the standpoint of regeneration, almost equally important, is how, in the context of a highly differentiated body, you can go back to producing something that really is very reminiscent of an embryonic structure and follows most of the rules, but yet it can't do it unless it's connected to the body.
Right.
So there's the aspect of integration of an embryonic or an embryo-like system to the remnant of the adult mature configuration. And that's something that people really haven't even given much thought to. One of the interesting things about that is that this phenomenon of regeneration of muscle in mammal is what I call tissue regeneration. And the regeneration of a whole hand, for instance, is called epimorphic regeneration. But there's an interface between the limb bud or the regeneration bud or blastema and the remaining limb stump where tissue regeneration occurs. Mm-hmm. So we have this sequence of adult tissue regeneration that doesn't follow the normal morphogenetic rules. Mainly it works on the basis of physical characteristics. And then you have something that follows most of the rules of an embryonic limb bud. And how those all come together is something I have not even seen a reasonable hypothesis as to how those integrate.
Going back to earlier development, do you have a similar problem of trying to coordinate everything that's happening in adjacent tissues so you get a cohesive whole?
Yeah.
It's a very difficult problem.
Yeah, it's really it's an integrative problem.
Yes.
And I mean, we've done such, not I, but the field has done such a marvelous job in dissecting the pathways leading to the formation of individual structures or organs, and now putting them all together is going to be one of the next big challenges,
Definitely
I think.
a big challenge for the future.
Yeah.
Okay, I'd like to switch gears at this point. We've been talking about Bruce's career and his role in understanding limb and muscle regeneration. But another life that Bruce has is he's an avid fisherman and is very interested in fish biology. In fact, has a very nice book on fish biology. Would you like to talk a little bit about your activities in fish biology?
Yeah, I'd love to. It started out when I was 5 years old, and I was fishing with my uncle in a lake in Wisconsin, and I caught about a 5-inch sunfish, and that automatically got me hooked onto fish. And I've been an absolute nut about fish and fishing ever since. And when I was in college, I was completely dedicated to becoming a fish biologist, and I put my way through school working for what at the time was called the Minnesota Conservation Department as an aquatic biologist. And then I spent a summer at the University of Georgia's Marine Institute, where I was working with a fellow. I was a technician in the lab where they were looking at the intermediary metabolism of a salt marsh. So I spent my mornings collecting filler crabs and then looking at oxygen consumption and respiration in the afternoon. But before and after work, I spent all my time collecting fish. Well, then when I got to Cornell, of course, then I was officially doing fish biology as a graduate student. Sure. And in the intervening time, most of my fish biology consisted of actually fishing, but doing a lot of observational biology while I did that. And then in the mid-1980s, I began writing articles for a fishing magazine called In Fisherman, where I would combine serious science with fishing. And the object was to let fishermen know that if they understood how fish operate, they would catch more fish. Mm-hmm. And I'm still doing that kind of thing now.
Is that theory true?
It actually it is.
The theory?
It definitely is, yeah. And then after I retired, now I've gone back to my childhood, so to speak, scientific childhood. And I'm now conducting a study on the growth of northern pike in an isolated Minnesota lake not far from our cabin. So part of the burden of doing this study is having to go sport fishing 2 days a week and catching 40 pike a day and tagging them and taking fish scales for samples. And then in the wintertime, I'm poring over data sheets and aging fish scales in my office. And this is a 10-year project, so I'll be in my 80s by the time this is done. I can see me pulling up nets and water 38 degrees or 36 degrees when I'm 80 years old.
Well, you're a tough guy.
We'll see.
You can take the cold.
Yeah.
Well, since you are such an avid fisherman, I can't resist asking the question or putting it this way. Tell me a fish story. You must have a fish story.
Oh, I've got some good ones, yeah. Actually, some people think that a lot of my science is a fish story, but that's another issue.
Or a fishy story,
Yeah,
I guess.
but I think some of the most interesting fishing I had is when I was on Sapelo Island when I was working at the University of Georgia Marine Institute. We used to go fishing for stingrays, and these fish were about 7 feet long and weighed maybe 150 pounds. And catching one was like hooking onto a Jeep, and I would periodically I'd land about one out of every 10 that I hooked. But one of my partners in the graduate student dormitory there didn't believe me, so one morning I took him out fishing, and about 5 minutes later I heard this voice, "Help!" and I saw this guy being dragged out into the ocean up to his chest in water by a stingray. So at that point he was a believer.
He was a believer.
Yeah. Then I guess my other fish story occurred a couple months ago. I was working what I call industrial fishing on this pike project, and the last fish of the day I caught, it was a 20-pound pike, which is the biggest pike I'd caught in my life.
Nice fish.
And that evening I went out to fish for walleyes, which is what I do for recreation, and also walleyes taste good. And my first fish that evening was a 13-plus pound walleye, which is extremely large for a walleye. So that's
How big is that? I mean, in length?
Oh, about 33 inches.
33 inches?
Yeah.
Oh. And what about the pike?
Oh, maybe 38, 39. Yeah,
Those
so
are big
they
fish.
get pretty big, yeah.
Wouldn't want to meet them in a dark alley.
Yeah. No.
Very big fish.
In fact, it's amazing because pike have such big teeth, and you try to avoid getting eaten by the pike if you bring them into the boat. And last spring there was one time when we were netting fish, and the smallest one we got was a pike about maybe 12 inches long, and somehow or other my finger got in its mouth and it bit my finger. And they must have an anticoagulant because the whole morning my finger was bleeding, and every single fish scale envelope we had was covered with my blood, not the fish's blood.
Dangerous sport.
Yep.
I want to go back for just a second to your graduate school experience, because you said something I didn't know about you, and that is that your advisor died while you were a graduate student.
Yeah.
So how did you complete your graduate career at that point? Did you have others who stepped up to the plate, or you just did things independently?
I just sort of did things my way, to quote Frank Sinatra, who I'm not horribly a fan of. But no, my advisor was
Please don't sing.
No, I can assure you I won't. My advisor, who was a reproductive endocrinologist, died after about 3 months when I was in the lab, and the rest of the department, I was at Minnesota at the time, was completely focused on diabetes research. But fortunately, the chair of the department, who had a lot of money, realized that I really knew what I wanted to do. So he says, "Don't worry about money. You can, basically inherit the lab and do your own thing." So the whole time I was in medical school and in graduate school, I was running my own laboratory, and it was a little odd because I really, number one, didn't know what I was supposed to do, so I ended up taking my qualifying exam after I'd written my thesis. And also it was convenient because I was one of the first people who'd been on the MD PhD program, and I was at the time registered in 2 other colleges besides the medical school, and they said, "Well, you can't do that." And I said, "Well, sure I can. This is a new program." So I mean, from my perspective, it worked out well. And finally, they had to have somebody sign my thesis. So we got William Feltz, who was a bone biologist, who was my official advisor, who signed my thesis. But I literally had nobody. And I think, you talk about mentoring these days,
right.
Totally unmentored, and I think maybe I still am, for all I know.
So I guess you still can succeed without mentoring. I always wondered if you could learn anything without objectives. I suspect you can. So the other thing I'd like to go back to is this time when you were in Russia and in Czechoslovakia. In the U.S., of course, we heard a lot about the KGB. And I'm just wondering if you had any experiences with the KGB or the CIA after you got back to the States.
Yeah, many.
Many.
Yeah, I mean, part of this was in the era of the Cold War, when they had these basically diplomatic games going on that cost enormous amounts of money. And one of the things the Russians always tried to do was to compromise visitors. And my being there as a single young scientist, I'd be a perfect target. So many times I had people approach me with various things. And I think the first instance I can think of was when I was in Leningrad. I'd only been in Russia for a couple weeks, and I could understand a little bit. I couldn't speak very well. But I was getting calls constantly in the evening in my hotel room, and I'd answer the phone, there'd be nobody there. And one evening, this must have been around, it was late December, maybe close to New Year's Eve, I got a call, and there was somebody at the other end, and I finally was able to understand the voice. And they said, well, could we get together? And I thought this was some kids that had been trying to sell me Russian medals, basically, for ballpoint pens, that sort of thing. So I made arrangements to meet whoever the caller was on the street below the hotel at 11 o'clock at night. Well, I got down to the street at 11 o'clock at night, freezing, and all of a sudden from across the street came two young women wearing leopard skin coats. And I realized they probably weren't selling ballpoint pens or Russian medals. So we agreed to go to the only nightclub that was open in Leningrad at 11 o'clock at night. So we sat there for a while, talked. They claimed that they were French majors, but my French is terrible, but it was better than theirs. And finally, we were trying to determine who we were. And so one of the girls took my Russian-English dictionary, and she found the word Virgin Mary and said, "Niet." And by the time that evening was done, I think I had made arrangements to spend the next night with her, but fortunately that morning I was supposed to go down to Tbilisi, Georgia, and I was scared to death that the fog would keep me in Leningrad. But that was one of the first instances, and I had times when a fellow at the Bolshoi Ballet was sitting next to me wanting to hold my hand and have supper with him in the hotel afterwards, and I had people wanting, well, the same guy actually I met half a dozen times, either exchanging currency or wanted to know if I like girls. One time he brought me into the apartment building where Khrushchev lived and took me into a 5-room apartment that was floor to ceiling covered with icons, and offered to let me have any icon I wanted if I'd give him my suit. Well, this is again a classic Russian play, and of course I figured it wouldn't be a good idea since the exchange program was semi-diplomatic, so I was tempted at that one. But then I ran into him at the Bolshoi Theater selling hawk tickets, and the last time I saw him was the last day I was in Moscow. He was in a kiosk selling something or other, but this sort of thing happened all the time. And we got to the point where it was pretty easy to identify KGB guys. And when I would come back to Ann Arbor, because I went to Russia and Czechoslovakia many, many times, the morning I got back, I'd have either a CIA guy or an FBI guy or both sitting at my office door. And one time in particular, there's an FBI fellow who's a neighbor of ours who was basically, I forget what we were talking about, but anyway, I told him that there's a really good way of identifying KGB agents, and I described the kind of leather jackets they wear and the clothes they had, and he says, you know, that's funny, because every time a Russian delegation comes to Ann Arbor, there's always one guy dressed like that who disappears, and we never see him until the delegation is leaving. But he sort of forgot about that, and about 1 or 2 trips later, he came back and was asking me something. He says, you know, Bruce, there's a fantastic way of telling KGB agents. They wear this sort of leather jacket. He forgot that I had told him about it in the first place. But also on our end, I used to have a few Russian visitors, and one of them was a fellow named Viktor Midashov, who was spent, I think, 4 or 5 months in my lab, and he was at our place over Memorial Day, where I took him to the local Memorial Day parade. And another FBI agent who was a neighbor of ours and a good friend of my wife's, and this other fellow were both there wearing Bermuda shorts looking at the parade. And my wife brought Victor to the FBI guy and said, I'd like to introduce you to Victor. And he says, Gene, please, I have a wife and a kid in a nice house. Don't introduce me to the Russian. And the rest of the morning, we had this scene of 2 FBI agents hiding behind trees every time I walked by with Victor because they didn't want to be even seen close to him. But there were a few cases where Russians have actually been harassed by people on our side. So it was mutual, definitely.
It was mutual, definitely. Well, Bruce, at this point, we have 2 options. One option is to sit here in the studio and continue with some more questions or any comments you want to make. The other is to go out and enjoy that Arizona sun.
I like option number 2.
Okay. So at this point, we'll end. I'd like to thank Bruce very much for sharing his insights with us and our audience for listening to this interview. It's really been a pleasure, Bruce, to talk with you today. And I really want to wish you every success now that you're retired, and hope that you're very much enjoying your life, and wish you every happiness.
Well, thank you very much, Gary. I appreciate that.
Yeah, okay. My pleasure.